Now that we have looked at ligand binding curves and the O2 binding curve for myoglobin specifically, we can use this information to tell us information about the physiological function of myoglobin. So the question we want to answer in this case is, would myoglobin be a good O2 transport protein? Or in other words, would it be good at picking up oxygen in the lungs where the partial pressure of oxygen is high and then deliver it in the tissue where the partial pressure is still lower than the partial pressure in the lungs? And so an estimate of the partial pressure of oxygen in the lungs is 100 torr. If we look at our ligand binding curve, we see that the axis only goes from 0 to 10 torr. And so what that means is 100 torr is way off scale. And we also know that within the 0 to 10 torr, we've already saturated most of the binding sites on myoglobin. So that means at even higher partial pressures, we're still going to be at saturating conditions. In this case, all that we want to have happen is that myoglobin is able to pick up oxygen. And in that case, this protein would be very good at picking up oxygen at the pressure of oxygen in the lungs. But remember, in order to be a good O2 delivery, that O2 also needs to be able to dissociate from the protein. And ideally, what we're looking for is at the partial pressure of oxygen in the tissues where the oxygen is being delivered, that pressure should lie somewhere within the curvature of the graph, near where the KD is, so where theta equals 0.5. However, an estimated partial pressure of oxygen in the tissues is 30 torr. And still, again, in the same figure, we know that 30 torr is again way off scale, so way to the right of 10. And so that means myoglobin is still saturated with oxygen, even at 30 torr. And so what that means is that oxygen would never be dissociated in the tissues. It would stay bound to myoglobin. And that's not what we need in order for us to have a good O2 delivery system, for a good O2 transport protein. So that's consistent with the use of a different protein, hemoglobin, in order to transport oxygen from the lungs to the tissues. So what do we need to have then in order to have a good O2 transport protein? So again, this black trace on the top and this figure on the right is the high affinity state, which is very much like the binding curve for myoglobin that we just looked at. What if we lowered the affinity? What if we just lowered the KD? Would that help? So let's say we just lowered the KD. And here's a curve on the bottom representing a myoglobin with a lower KD. And so what we find here is, well, now the curvature of this curve is within the partial pressure of the tissues. So O2 would definitely be dissociated in this case, and that would solve part of our problem. However, as we see for this partial pressure in the lungs, which is shown by this red stripe here in this chart, we don't get very good saturation of the sites. Only about half of the sites in this case would be occupied with oxygen. So that means while it might dissociate the oxygen well in tissues, it wouldn't bind O2 very well in the lungs. And so we need to have both of these functions in conjunction. Hemoglobin can do both. So hemoglobin, you see, has a different curve entirely. So instead of a hyperbolic curve consistent with the ligand binding curve equation that we've been looking at for myoglobin, it has what's called a sigmoidal curve. So that means that it has basically an S shape in the curve. And so at low oxygen concentrations, there's a lower affinity for oxygen by hemoglobin. But as you get high, this starts to increase rapidly, and you get saturation at higher O2 concentrations. And if you compare the O2 binding curve of hemoglobin to the partial pressures of oxygen in the lungs and in the tissues, you see that hemoglobin is really well designed for being able to pick up oxygen in the lungs at 100 torr and then deliver oxygen to the tissue at lower partial pressures of oxygen at 30 torr. And that's what makes hemoglobin a better protein than myoglobin as an O2 delivery protein in the blood. So what actually provides this function? What is causing this difference in these ligand binding curves and the different function of myoglobin versus hemoglobin? And so we know that ligand binds at the ligand binding site. So you might think, well, there are differences at the ligand binding site. But actually, if you look at the protein structure, so that here on the left is a crystal structure of myoglobin, and you can see, again, myoglobin is a monomer, and you can see the fold of the myoglobin subunit, or excuse me, of the myoglobin monomer, and you can see the heme cofactor that's inside of there. And then compared right next to it is the beta subunit of hemoglobin, just for comparison. And you can see just by looking at the peptide backbone, so looking at the alpha helices and their arrangement, the structure of the protein is very similar. And in fact, the placement of the heme is also very similar, almost identical. And so the difference in the structure of the two proteins has very little to do with what's going on at the ligand binding site. For further proof of this, we can look at the amino acid sequence. So remember, primary structure, right, dictates different tertiary, secondary and tertiary and quaternary structures, and also, and that may give us a clue as to what's controlling the function of this. But if you look in these, so what we do is we do what's called a amino acid sequence alignment. And so this is a computer generated program which aligns the amino acids based on their similarity to each other. So we have three amino acid chains shown here. One is the alpha subunit of hemoglobin, the second one is the beta subunit of hemoglobin, and the third is myoglobin. So where the letters are red, these are called identities. So that means that in all three proteins shown here, the amino acid at that position is identical for all three. And so there are many identities, and so what this tells you is that these proteins are very similar, even in amino acid sequence. And so what we find is really important amino acids that have identities are known as conserved amino acids. And these are usually really important for the function of these proteins. And so what we see is the proximal histidine, again, which ligates to the heme iron at the ligand binding site. And then we also have the distal histidine, which is important for forming a hydrogen bond with the bound oxygen in order to stabilize the O2 binding. But it doesn't give us a lot of clues otherwise as to what's going on here. And for that, we need more structural information to figure out what it is exactly that is causing the differences in the functions. What is the structural differences between myoglobin and hemoglobin that allows for the different functions of these two proteins. And remember, at the beginning of this lecture, we discussed what's a very critical difference between this. Myoglobin is a monomer, whereas hemoglobin is a tetramer. And that's actually very key towards being able to allow for, instead of a hyperbolic binding function, O2 binding function, which is observed by myoglobin, and converting it to a sigmoidal function, again, which is important for the function of hemoglobin as an O2 transport protein from the lungs into the tissues.